Hello, everyone. Welcome to today's live broadcast, Explore Precision Medicine, QCI Interpret to Elucidate Precision in Breast Cancer Targeted Therapy. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by QIAGEN Bioinformatics. QIAGEN Bioinformatics is powered by CLC Bio, Ingenuity, and Biobase. The company offers bioinformatics software tools for next-generation sequencing, NGS, data analysis, and interpretation. Its solutions are designed to be universal, so customers can mix and match the technologies best suited to their needs. For more information, please visit www.QIAGENBioinformatics.com. Before we start, there are a few instructions. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too, and you can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right, or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Julie Deschenes, MBA, PhD. Dr. Deschenes is the Manager for Advanced Genomics at QIAGEN Bioinformatics. She is a strong customer advocate working closely with customers and partners, identifying insights in their NGS and expression analysis data and facilitating the implementation of their clinical decision support software. Dr. Deschenes completed her PhD in biochemistry and molecular biology in 2008 at the Institute for Research in Immunology and Cancer in Montreal. She studied breast cancer by looking at the transcriptional regulation of estrogen receptors to the DNA through long-range regulation by estrogen. She has been an author on numerous peer-reviewed articles and spoken on a variety of high-level panels, symposiums, and conference sessions. Dr. Deschenes recently obtained an executive MBA on market strategy for a next-generation sequencing clinical reporting software from the Würzburg Universität, Germany, while supporting the market entry of QIAGEN Clinical Insight Interpret. Dilhan Weeraratne, PhD, will answer attendees' questions during the Q&A session following the presentation. Dr. Weeraratne is a senior scientist working in advanced genomic support at QIAGEN Bioinformatics. Specifically, he is a cancer genomics scientist invested in leveraging the explosion of genomics data to transform cancer treatment and patient outcomes. He has more than a decade of experience in cancer genomics and precision oncology, both in academic and industry settings. Dr. Weeraratne obtained his PhD in neuroscience and molecular biology from the University of Vermont in 2007. He then joined Harvard Medical School and the Broad Institute of MIT to do postdoctoral research in medulloblastoma genomics. His work was instrumental in defining the current World Health Organization accepted molecular subgroups of medulloblastoma. Following his postdoctoral work, Dilhan transitioned to biopharma and worked at leading diagnostics companies, including Thermo Fisher and N of One, focusing on clinical interpretation and annotation of biomarkers in solid and hematological tumors, clinical reporting, and developing pharmacogenomics biomarker knowledge bases. Their complete bios are found on the LabRoots website. I will now turn it over to Dr. Deschenes for her presentation.
Thank you, Judy. So for today's presentation, Dr. Weeraratne and I, ovarian scientists, we worked together in building this presentation, which we hope will illustrate how clinical decision support software, such as the QIAGEN platform here, allow laboratory to address challenge in genomics. So without further intro, let's get started. So during the presentation today, we will essentially start with our QCI platform, or QCI Interpret. And then we will go into the breast cancer facts. So as you know, breast cancer is a complex heterogeneous disease, which each subtype characterized by distinct clinical feature and molecular alteration. The advances in NGS have presented unique treatment opportunity beyond stratification of patient by hormone receptor and epidermal growth factor receptor status. So the presentation today will demonstrate how tool like QCI can help laboratory in oncology. So to illustrate that, we have essentially selected 5 different cases. 2 cases coming from the TCGA atlas on the somatic side of the breast cancer, 2 cases coming from the hereditary side of breast cancer, and 1 last case which represents essentially triple negative example. And then after going through the different cases and how you can leverage QCI with the interpretation of those cases, I would conclude with a summary. And the learning objective will be for you to see the utility of QCI Interpret in identifying molecular alteration, see how treatment and clinical trial decision support can be done by using tools such as QCI, and then see how we can demonstrate the richness of the QIAGEN knowledge base using this tool. Before we get started, I have to put some intended use statement about QCI. So QCI is an evidence-based decision support software intended as an aid in the interpretation of variant observed in genomics alteration. It is not intended as a primary diagnosis tool by physician or to be used as a substitute for professional healthcare. When we think about NGS and when we think about the challenge in the clinical genomic testing, there's a lot of different challenges. If we start with the first one, we can see that the first challenge is often the scalability and accuracy of the interpretation and reporting. In essence, you have a lot of different data. You have a lot of data to analyze at any given time. So this is one of the challenges that can be addressed and will be addressed with a tool such as QCI. The next challenge that clinical laboratory has is also mitigating the risk with their test menu expansion. So often what may happen is that a laboratory will start testing one type of case, and then an additional disease area, and another one, and another one. Therefore, not only you need a structure that would be scalable, allow you to look at all these different test menu, but also making sure that this is done in a way where you can really have your test menu that expand and provide rich information similarly across all of them. Another challenge is the continuous improvement that you may have while running your lab into making sure that you are getting good operational efficiency, that your turnaround time is as quick as possible, and then you're keeping your production costs. And then finally, what is also important with genomic testing is also making sure that you're continuously learning about the new science, about the new drug, about new clinical trial, and so forth. So you are able to bring essentially a lot of different information all the time. So these are challenges that QIAGEN is trying to address with this solution. So the end result of that is what we call the actionable report, which is what essentially clinical laboratory often have to do, which is starting with running the test, make the interpretation, and answer 3 basic questions. When you do that interpretation, you want to know, as a variant scientist or as a laboratory director, is the observed variant real, for example? Is it clinically relevant? It doesn't matter to have that variant, essentially. And more importantly, is it actionable? So are you talking about a variant that doesn't carry any actionability, therefore may be less important into the clinic? So these are the type of questions that in today's presentation, I will use the 5 different case studies in breast cancer to illustrate the benefit and the breadth of the knowledge of QCI and how it can help answering those questions for laboratory. But before diving into the case study itself, it is important to realize that clinical decision support tools are only as good and accurate as they are built on a strong foundation. So the QIAGEN knowledge base is a solution that has been done in making for almost 17 years now, where we essentially have two dynamic. From one side, we have the content acquisition dynamic, which is essentially been for more than 17 years the QIAGEN bioinformatics area throughout the different acquisition has been curating a lot of variety of information from basic research, clinical information, linking this to treatment and so forth. Now that content is a lot of information to take in. Therefore, what you also need is making sure that you support all that content with a structure and ontology that allow you to essentially making sure that you can connect the different dots. So making sure that you can connect things like patient phenotype with disease mechanism, with cellular mechanism, that sort of thing, to make sure that you have the whole story and the whole picture for your particular variant. And this is what is at the essence and the core of the QIAGEN knowledge base. And the QIAGEN knowledge base is essentially leveraged by the QCI platform that we will see during this presentation. We are just off of the the October month, where October was the awareness month for breast cancer. We're now getting into November and so forth. But it is important to realize that breast cancer is an important and still an important disease area. It is just in the United States touching about 1 of 9 women, or 12% of the women will get breast cancer at some point in time in their life. There is a lot of different new cases a month for breast cancer. The survival rate is good. There is a 5-year survival rate of 90%. However, breast cancer can quickly metastasize in different other parts of the body. Therefore, it is really important to be able to have this different tool to address that throughout the whole continuum of different type of cancer. There's mainly 2 different types of cancer, and as you probably a lot of you are familiar with, a lot of the way to classify is to classify them with their biomarker. So many breast cancer, for example, will express HER2 or ERBB2, estrogen receptor, progesterone receptor, and these biomarkers are used, in targeted therapy for a different patient. However, there's also a component where about 15 to 20% of the patient will be triple negative for these biomarkers. So what we thought we would do during this presentation today is we'll essentially use, we use TCGA published cases. These are real patient cases that have been published, and we're using them to illustrate how a solution like QCI Interpret can allow you to address all the challenge that you may have in the clinical laboratory. So the first 2, as I mentioned, will be about 2 different cases of somatic cases where the driver mutation in those cases are more around PIK3CA, P53, and so on. Then we will take a look at an example with the BRCA1 driver mutation, as well as an example with the BRCA2 driver mutation. And then finally, we will take a look at a particular case, which was triple negative with regards to the classic biomarker, where the driver mutation is most likely more around MAP2 kinase. So, and that is, that is allowing us essentially to show you the breadth of the different type of information you can get within the system. So today's presentation, I will be, for simplicity, I will be using a slide presentation, but throughout my presentation, what you will see is a lot of different screen captures. So the QCI tool is a cloud-based solution that essentially will allow you to send or submit within a NEPA compliant way, obviously submit the variant file information, and then see all the richness of the information around a particular case, and then look and potentially report out the information based on the case. So today I will use a slide to do this exercise. but this is a web, a cloud application that can be really onboarded and spin off and used by customer really quickly. So let's dive into this specific case. So the first case is the case of a 43 years old Asian woman. When she came, based on the information, she was presenting with a stage 2A breast invasive ductal carcinoma. Her status was HER2 negative, PR and ER positive. She had BRCA1 and 2 variant as well. However, based on the case and based on the information available, the driver alteration in that particular case was most likely a PIK3CA and the P53, and that's what I will spend most of my time discussing about. So now let's essentially go and look into the data. So once what we did is we essentially submitted the variant call file, the information for that particular case into the system, and what you see when you launch the interface is some sort of a dashboard, and I know it might be really small from your side of the computer, but what you essentially have is a dashboard that allows you to see 3 different sections. One is the variant list, which is essentially all the different variant that is in this case. So it's important to know that here, for the interest of this case study, we are presenting only the somatic or cancer important variant. We have not, these were all genome, all exome samples, so we did not load all the different variants, so we kept only what was relevant in the context of oncology. But what we see here is essentially my dashboard that is presenting to me 5 different variants. 3 of them are color-coded as red, which mean pathogenic. So these are variants that are obviously carrying some pathogenic information. So there is, in particular, PIK3CA, which we will take a look a lot during this presentation, and then there's as well P53. You can see for each of those variant, we have different markers or different flags that essentially allow the end user to see actionability information. You can see things such as treatment, clinical trial, and so forth. So you are really able to have an idea quickly about what might be relevant or actionable in these particular cases. And I want to point out to you these BRCA1 and 2 variant. They were in gray, which means that they're carrying an uncertain significance. And what does that mean is they're not necessarily actionable based on all the computed classification that went through them. They're most likely not the one that is driving pathogenic importance for that particular example. So if we dive a little bit more into the information that you can get into this dashboard, is you have information about the specific variant that is highlighted. So I highlighted PIK3CA within the context of cancer, so I can see things such as what is a particular variant, what is the protein translation information. So these are key information that a variant scientist will need to use in order to be able to make their decision. There is things such as somatic frequency, population frequency, Allen fraction, and so on, that are also information that might be useful when you address and you look at the cases. But what is most importantly is to get into the detail for each of the variants. In some cases, you may say, especially if you see those every single day, you may say, I know this is the driver variant, and you move on and you do your reporting. But in some other cases, you may want to go and dive into the specific information. And that's what I will do here with PIK3CA. Let's dive into what we call the variant detail. The variant detail is essentially a section that is divided in a lot of different subsections that allow you to see specific pinpoint information. There is obviously things such as the variant detail, the assessment information. How can QCI say this particular variant, for example, is pathogenic? That sort of thing. There is things such as the treatment information, reported clinical case count, and so forth. So if we first take a look at the variant detail, the variant detail allow us to see things such as the chromosome, the position. There is a few different links here that you can see. So if, for example, you're coming at a cross and you want to know what does PIK3CA do, is it really relevant usually in breast cancer or in other type of cancer, you can quickly go and look at different type of information, such as the gene review databases that allow you to read about that specific gene, the health condition for that specific gene, and so forth. So quickly, the interface is allowing you to navigate through all the specific information that you may need to decide about this particular variant. If we take a look now into diving into the specific information, where is the variant? This type of information is represented in the section that we call missense effect on the protein, or the effect on the protein. So here what you have is where, essentially, where is the variant? So the variant is where this black line is. So this variant here is in the exon 10 of PIK3CA. It is in this particular section that we have here in yellow, which represents the critical domain of the protein. And throughout the interface, you can see these little red bar that will allow you to know whether or not the variant is in a hotspot. And we can see, if we take a look at what is around this variant, we can see that essentially this particular variant is indeed in a hotspot. So based on the literature evidences, there is a lot of variant in that region that deemed to be pathogenic in the literature. There is also information that you can see around gain of function. So if I click on the little I, and this is throughout the interface, I will get information. In this particular case, I have information about why is this variant conferring a gain of function. So this variant is conferring gain of function based on the literature information. So I quickly can see why does the software tells me it's a gain or loss of function, for example. So it's really allowing me to see the rich contextual information there. Now, you may say, in some cases, you may say, well, I want to know more about what is actually the literature. Is there clinical case in the literature that is talking about that specific variant, and are they in my specific disease of interest? So when you look and open the reported clinical case count, you will have this some sort of diagram that will represent for you the type of literature evidences that QIAGEN has curated. And what I mean by that is QIAGEN really has employed that their sole role is to curate literature and making sure that they're grabbing contextual information around the specific variant, ethnicity when it's available, and that sort of thing. So all of these different icons represent a piece of information that we have curated manually following a strict protocol. And it might not be that obvious in the broadcast, but you can see that there is different the shape are a little bit different. So essentially the interface allows you to see whether these literature evidence are coming from somatic information, coming from hereditary information. If it's hereditary, is it coming from segregation type of cases, and so forth. So you can really grasp an idea of the context. And here, quickly, without even diving into the information, I know that there is, a huge majority of the clinical case in the literature today that are around somatic for that specific variant. Now, if I want to know more and dive into the specific information, I can click the open option, and then it will open for me all the specific cases. So here we can see highlighted here that this is a publication that was published earlier in September. So since we are releasing content information on a weekly basis, if our curator has curated something last week, you will be able to see it this week into the system. So here is the case of a literature that is talking about that particular variant in primary breast cancer and also in metastatic breast cancer, where other cases have had that same PIK3CA. And so it's not just, another type of cancer like ovarian cancer, that sort of thing. It's really, these are primary clinical case in the literature that are around breast cancer. And you can, the variant scientists at this point, if it feels and deems that the particular literature evidence is relevant, then they can quickly click on this icon here and add the information to the report. So this little square icon that I just highlighted in yellow is essentially a way to click in the software to say, I want to put this in my report, or I don't want to put this in my report. So you can quickly add the literature evidences into your report, which is what exactly I did for this particular one that we can see here. So you can essentially add this. So as you review the information, you can already, without noticing, building your report. If you want to go and dive into the particular article, one thing to keep in mind is everything in blue here, everything highlighted in blue in the software will allow you to be directed to the primary information. So here it will be PubMed, for example, which is exactly what I did. So you can click on that blue link and you will get into the primary literature evidences. And if your institution has access, then you can directly go and then access the publication. So if we, now we've seen that with the PIK3CA, we can learn about what is PIK3CA in the context of we can access information about whether or not that particular variant has been seen in other clinical case around breast cancer, which we have confirmed. In many laboratories, one of their goals is also to provide treatment, either treatment or clinical trial. And this is what we can see here. When you open the treatment information, you can essentially access an interface that allows you to dive into the treatment and clinical trial information based on whether the treatment or the clinical trial correspond to the exact variant, which there's none in this particular case. We don't see any treatment, we don't see any trial, or whether you're looking at the entire gene. And you can ask that question within your specific cancer, which is what I did here in breast cancer, or you can also go and see the information for any type of cancer. So if we dive into that specific information and we look at any type of treatment or trial within breast cancer, that's exactly what I have here. I can first notice that there's still no treatment. So there's no approved treatment at this point in time for PIK3CA itself. But the bottom part is the clinical trial. So here we can see a few different clinical trial, and unfortunately I cannot scroll because you would have been able to see all of them. I had only 4 in my screen capture, but you essentially have different trials that are currently enrolling. So at this point in time, or at least last update, which we do every week, typically on Thursday, then you have a clinical trial that are currently enrolling. You can narrow it down, which we did here. We kind of kept this within the United States for the purpose of the case story, but you can narrow that down to the primary location where the physician is, the ordering physician, for example, is, and that sort of thing. So if we dive into the treatment information and we take a look, and again, I do apologize because it's a little bit small on the screen, but you can see for the first treatment and trial, actually, it is a PIK3CA where ERBB3 is negative. So it's a case of a treatment where PIK3CA mutation is present, ERBB2 is negative, which corresponds to this particular case, and estrogen receptor is positive. However, when we take a look at the description and the study of the title, I highlighted in blue here that in this particular criteria for enrollment is this trial is open to postmenopausal women, which was not the case of this particular case. Therefore, this trial in this particular case would not be applicable for that particular TCGA case. However, if we look into a tiny bit lower, you essentially have the phase 3 PIK3CA inhibitor that have clinical case that are, clinical treatment that trials, that are currently enrolling, for which this particular case could be, applicable. So essentially, Dilhan did made them as reportable. So these are clinical cases that can then go within the report of that particular case. And once the variant scientist and the laboratory director have done reviewing all this particular information, then you can essentially go and dive into more detail why the assessment, for example, was done, and then generate a report. So here we can see the different criteria or rule that made this PIK3CA assessment possible for pathogenicity. So you have all the different criteria. If the laboratory do not agree with it, they can always change the different criteria on the fly. So it's allowing you to essentially turn on or off on the computed classification if it's something that is not applicable for a given case. And once you agree with the assessment and you are ready to generate the report, you essentially go and then you click on the last tab, which is Review and Report. And that allows the variant scientist or the laboratory director to put their overall interpretation on the case, and also allow them to put overall interpretation on the different variants that you have. So QIAGEN already provides some sort of text, but it allows you to enrich, add contextual information around the particular variant, and even save it for reuse next time. And if we think about the biology of the reality of this case, in this particular case, what the interface allow us to look at is that the BRCA1 and 2, while people will think, oh, breast cancer, BRCA1 and 2 type thing, they were not pathogenic in this particular case. PIK3CA is an activating mutation in the hotspot region. It's eligible for clinical trial, a different few clinical trial in breast cancer for PIK3CA. And what we have not looked at, there's also potential clinical trials for the P53 mutation that could be done. So when you go through the whole interface, you can essentially make these type of conclusion and finally generate a report. And the report engine can either be an electronic information that is in your within your laboratory, or like this, which is essentially a PDF that will contain all the information about the specific cases. So now, if we take a look, when we were doing this case study, one of the things we were wondering, and one of the questions I was asking Dilhan, is what about male breast cancer? So can we take a look at an example of male breast cancer and see, is it different? Is it the same? And that's what we have done. So as you, for the next case, as you may know, male breast cancer is really, really rare. There's about 2,000 different new patients in the United States that are newly diagnosed every year in average. However, what we will show here is essentially the etiology of breast cancer transcends gender. So what we will take a look at when we will look at the variant is that PIK3CA is also, it's a different variant, but is also a gene that has carry the driver alteration in this case. If we think about the case itself, it's a 68-year-old Hispanic gentleman that was presented with a stage 2B luminal B-invasive ductal carcinoma. He was ER2 negative, ER and PR positive. The BRCA were negative in this particular cases. The histology was really similar to female breast cancer in that particular case. So we did a similar exercise where we essentially took a look at the variant that were relevant for oncology in this particular case. And we there was not a lot of them that were highly relevant, but there was one that was striking, which was the PIK3CA variant again. It's a different variant. It is also a pathogenic variant. As you can see, the profile of the literature evidence compared to the other one in these cases, we see that it's a little bit more half and half, where you have a lot of somatic clinical case in the literature, but you also have a good amount of the literature coming from something a little bit more inherited or individual type, but not necessarily a somatic cases. So these are type of information that we're able to gather really quickly. And when we go and we dive into the treatment or the clinical trial, it's a similar story. So we're also looking here about all the specific clinical trial around PIK3CA. And in this case, though, if you think about a male, a male is essentially somewhat could be considered as a post-menopausal situation. So it is possible that this first trial that we were looking at earlier could be relevant for that particular case. So that might be something that the laboratory director may want to investigate further in terms of the criteria, specific criteria of the clinical trial and so forth. So the interface is really allowing the laboratory director to have access to this information and quickly decide whether or not this is relevant. But in conclusion, when we think about this male, is while they are really rare cases, the disease etiology and the type of gene that are affected can be really similar, like we can see in this particular case. And in this one, it is also a PIK3CA activating mutation in a hotspot region. And potentially the same clinical trial could be, with PIK3CA inhibitor could be of interest for the laboratory director to put into the report. So the next thing we said, it's nice to have an interface that allow us to take a look at somatic cases, but what about inherited oncology? What about inherited breast cancer? What can we do with an interface such as QCI? And that's what we did for the next two cases. So we selected two cases of hereditary, one case coming from a BRCA1 driver mutation and a different case coming from a BRCA2 driver mutation. So this first one is a case of a 39-year-old African-American woman presented with stage 2B cancer as well in the upper left quadrant. She was HER2 negative, PR, and ER positive. The BRCA status was BRCA1 positive, no BRCA2 variant in this particular case. She, at the time of the sequencing, she was premenopausal in this case. And for some of you that are a little bit more involved with the particular disease area and so forth, BRCA1 are often more typical, not always, but more typical of a variant that will be in non-Caucasian individual. And that was kind of one of the questions we were having is whether when we look at the content or the information in the knowledge base, whether this was reflected as well. So when we uploaded this into the interface, we first confirmed that this BRCA1 variant, or this variant in BRCA1, was indeed pathogenic based on ACMG classification. This variant, when we take a look at the assessment, there's a lot of different criteria, such as null variant, a variant that is not present in any databases. There's also information about CAD information or effect of the gene and the gene product, essentially, that were of interest for this one. If we, when we opened the bibliography information, we have found one bibliography. So there was really one case in the literature at the time we did this, which is about a month ago. At that time, there was one particular literature evidence that was talking about it. So and that one was in affected cases. So we took a look at what this publication was about, and this is a publication that talked about that same variant in the BRCA2 gene, and it was observed in breast cancer, but in Lebanese. So it was inherited BRCA1 variant in the Lebanese population in this particular publication, something that was published in 2015, which was really interesting for us and comforting in a bit, where we could say, okay, there's really this notion of different ethnicity that can carry this variant as a familial inherited cases. So we were able to look at that information by accessing this publication. When we think about treatment and trial, there is some things that can essentially be done with regards to BRCA1 variant here. So there is the standard therapy, obviously, but there's also, when we think about therapies, in the case of BRCA1, there's olaparib, for example, that is currently approved by FDA, EMEA, and NCCN for ovarian cancer. So what we were interested in looking is to whether or not, so this is an approved drug in a different cancer, ovarian cancer, so not that far from breast cancer essentially, and the question was whether or not there's actually a clinical trial as well for breast cancer, for example. And that's what we took a look by looking at the clinical trial, and there is indeed, and I apologize again, but there's indeed different clinical trial with olaparib and others that are also for the, also open currently for the second generation of PARP inhibitor. So essentially in breast cancer. So essentially, the laboratory and the medical team could have two different avenues here. They could ask for derogation and go into trying to get a derogation to have a treatment that is approved for ovarian cancer for that particular case, or they could also enroll this particular patients in different clinical trials that are more relevant for breast cancer. So that's essentially the conclusion of this particular case. So which would be, to look into the second generation PARP inhibitor essentially as a clinical trial. So our next case was about hereditary BRCA2. So in the case of BRCA2, so we had a 51-year-old Caucasian female that was presented with a stage 3. She was premenopausal in her cases, and we essentially did similar exercise, and I will go a little bit faster here, but essentially we went into, what are the different variants. At the time, based on the computed classification, the BRCA2 was a likely path, so a little bit less stronger. There was some NKT in there, but the BRCA2 was more likely pathogenic in this particular case. And the assessment, when we were looking at the assessment, were just, a little bit less evidences or rule based on ACMG classification that was less strong, more moderate type of rule. Therefore, the computer classification couldn't tell us that this was, pathogenic for sure. It's likely pathogenic. So it's not as strong as the previous case we have seen. Obviously, if you take a look at BRCA2, it's a similar thing. There is indeed, the PARP inhibitor that you can also take a look with the BRCA2 variant as well, which is similar story than the previous case when you get into these type of treatment, because they are relevant for both the BRCA1 or 2. So it's again, that same endpoint with regards to the treatment, but you can really see how the computer classification can allow you to navigate through to the different variants and see whether they are pathogenic, likely pathogenic, and all of that is computed in a matter of a few minutes when you submit the file to the interface. And lastly, because for the sake of completeness, we wanted to, I was asking Dilhan, I said, but what about triple negative? What about those cases where there are no known, there are no carrier of or BB2, estrogen receptor, progesterone receptor, so they're really negative for that information. what type of variant there is in those type of cases. So in this particular one, it was a triple negative breast cancer case. It was a case of a 50-year-old Caucasian woman, and as you know, it's a little bit more important in those and critical because these particular women cannot even go to things like anti-estrogen, like tamoxifen, aromatase inhibitor, and that sort of thing. So that was one of the interests that we had by looking into that specific case. And when we took a look within the interface, we found variant in P53, but also a variant within the MAP2 kinase, which, based on Dilhan, or variant scientists essentially experience, it is most likely the driver mutation in that particular cases. So it's a MAP2 kinase and a hotspot mutation. There's also a loss of function with P53. When you take a look at the different treatment, there is different treatment, MAP2 kinase, there is for that exact variant resistance, but there's also therapy for MAP2 kinase where you have a specific treatment that might be available. There's also a lot of different clinical trial that could be used. So based on the information, this MAP2 kinase was an activating mutation. There is FDA approved that are off-label. There is also different clinical trial in earlier phase, though, in this case, and then there's also information that, from a treatment perspective, that you can get when looking at the P53 variant as well with the WEE inhibitor. So in summary, with a tool like QCI Interpret, it's allowing you to essentially see all the variants that you have in a particular case. And today we have to take a look at the breast cancer case, but you can certainly do that for the software is really launched for the oncology, so be inherited cases or somatic cases or solid tumor or liquid biopsy. If you're doing more blood type of investigation. The dashboard allow you to, the interface allow you to have the rich contextual clinical case count, access to bibliographic information to make your decision, access to treatment and trial information also to help supporting the decision of the variant scientist in the clinical laboratory. And finally, it also allow you to generate your report, add your overall interpretive comments with the information that is needed for the particular case, and then generate the report. So in essence, you can stay current with the QIAGEN knowledge base on a weekly basis. There's new information, the clinical biological type of finding, available status of drug, clinical trial, that sort of thing. It allows you to increase the throughput that your clinical geneticists and variant analysts can do for interpretation and reporting. It allows you to stay current with the industry-leading knowledge of information up to date, as I mentioned. It also allows you to improve your clinical decision-making by automatically applying relevant professional guidelines, variant classification, rules to deliver actionable and evidence-based information, and then it can be customized to get maximum flexibility of integration within your internal system, your laboratory policy. There's possibility of making this and have your laboratory director team or medical director team to essentially drive the policy that are applicable for each of the cases and the workflow that are applicable for your laboratory. And then finally, you also develop a private lab-specific knowledge base. So what we have not seen, because I haven't been able to really go in the interface itself, is that it's you can see whether you have seen this particular variant in the number of cases at every time in new cases. So I will conclude my presentation here, and thank you for being with us today. It was a immense pleasure for me to do this presentation, and now I believe we will open for questions.
Excellent presentation, Dr. Deschenes. Thanks for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to reach us today. Questions can be sent via the green Q&A button at the lower left of the presentation window. We'll get to as many as we can. And our first question is, thank you for the interesting case studies. The triple-negative breast cancer case seems like a great example of how precision medicine will help in the clinic. Can you elaborate more?
Sure. Yeah, thank you, Dr. Deschenes, for that great presentation. This is Dr. Dilhan Weeraratne. So this is an excellent question. So this is a classical example, fantastic example of how precision oncology or targeted therapy will probably be the only only available treatment option for this patient. As Julie mentioned, triple-negative breast cancer is pretty challenging to treat, especially because the patient cannot be treated with trastuzumab because the patient doesn't have a HER2 mutation, or anti-estrogens like tamoxifen for the absence of estrogen receptor and progesterone receptor. So typically in the clinic, what would happen is a triple-negative breast cancer patient would be treated with anthracyclines like doxorubicin or epirubicin, or taxanes like docetaxel or paclitaxel. But the problem with this chemotherapy is that there are a lot of side effects, there are a lot of grade 3, grade 4 toxicities. So especially in the case of doxorubicin, many patients have cardiac complications. So I'm pretty sure when these patients treating oncologists saw the clinical reports, he or she would have been really happy, because most likely this patient would have exhausted 2 or 3 lines of treatment, and at this point the patient's only recourse would have been hospice. But in this case, there was a MAP2K1 mutation, and this is like a really great example. I was very happy when I did the analysis where the patient could be treated with trametinib off-label. So off-label in the sense that it's approved for malignant melanoma in 2014. So and also there was another MEK inhibitor, cobimetinib, which was approved last year. So what the oncologist would typically do is, okay, there is an approved drug for this particular mutation in a different indication. So the oncologist can use the clinical information in our application and write to the insurance company a letter of medical necessity. So this would facilitate the doctor to obtain prior authorization from the insurance provider, because these targeted therapy is not cheap. Trametinib is about $15,000 a month. So when they get the insurance prior authorization, the patient could be treated with trametinib or cobimetinib, or alternately the patient could be treated in clinical trial setting if a trial is available close to the geographical locality of the patient and the oncologist. So again, this was a great example of a targeted therapy helping a breast cancer patient.
Thank you. Dr. Weeraratne, from a variant scientist / clinical geneticist point of view, how would QCI Interpret help a clinical lab? So most of the time spent by variant scientists is on the literature curation and variant interpretation. So we have done an internal study here at QIAGEN, and we have shown that QCI Interpret reduces the test interpretation time by almost 75%. The time that is saved by this can be used for secondary review, QA and compliance, and especially for scalability. So other clinical decision support products annotate variants using public databases like HGMD or ClinVar. But the problem is that they lack curated content from primary literature, and this becomes challenging, especially if the variant interpretation is tied to a CMG classification. And as Dr. Deschenes mentioned, the strong point of this whole application is the underlying knowledge base. Our knowledge base is curated by PhD level curators, and we curate things from primary literature, clinical
clinical guidelines, not only NCCN, like many different clinical guidelines and FDA and EMA drug labels. I think the other definitive advantage of QCI Interpret over other clinical decision support systems is that QCI Interpret assesses the entire genetic profile in its entirety, so matching combination variants or multivariants with therapy. So if there's one single variant, the therapy or the recommendations that would be given would not be on a particular variant, but the composite molecular profile of that patient, which is how a patient should be treated in the clinic.
What data sources are integrated into this tool? Can you list them?
So, it would be a little bit too long to integrate. There's about 30 different sources. So the way QIAGEN is building the knowledge base is we first for sure have what the typical databases that you would think, such as ClinVar, Cosmic, HGMD, databases. You also have, for example, if we think about guideline, you have NCCN, FDA, EMEA guidelines. You also have things such as clinicaltrial.gov that is integrated, that we curate fully, which means that we will not just integrate the databases, but we will also make sure that the status, the criteria match, as Dr. Weeraratne has said. And there's primary literature. So QIAGEN also has all the little diagram that I was showing. These are primary literature that we curate at QIAGEN directly. So we have our team of curators that will read these clinical paper, and they're primary clinical case in most of the cases. There's also databases when you think about protein function that will be used. There's also different protein predictor type of databases, CIF polyphens, that sort of thing that will also be integrated. There's a list about 30 or so of them, and we will be more than happy to have one of our support scientists reach out to you personally after and provide you the list and the correct, all the information about the different databases.
For genes that have multiple transcripts, which transcript do you represent on QCI Interpret? And also, do you have information on lesser known transcripts?
Oh, that's a great question. A question we get quite often from our clients. Yes, we do. So for genes that have multiple transcripts, the QIAGEN default transcript is the one we have most number of findings in our database. Typically, this represents the most clinically relevant transcript. So if two transcripts have the same number of findings in our knowledge base, we would assign the longer transcript as the default transcript. But this does not mean to say that the other transcripts are not available. Every single transcript can be selected via toggling at that particular section. So, for example, we have information for FGFR1, like many of you know, has multiple transcripts. So we have 9 different transcripts in our QCI Interpret application. For GNAS, another common gene where there are multiple transcripts, we have 7 different transcripts in our application. So if you are interested in a particular transcript, you can easily scroll down and select the transcript of interest.
Well, thank you. And for those, if we didn't get to your question today, we will follow up with you via email. I would like to once again thank Dr. Deschenes for her presentation and also Dr. Weeraratne. Do you have any final comments for us today?
Not really. I hope everyone has a great day, and thank you for being with us today. It was a pleasure for us.
Thank you very much.
Thank you both. I would also like to thank our sponsor, QIAGEN Bioinformatics, for making it possible to bring this presentation to you. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available for on-demand viewing through February 2017. You'll receive an email from us alerting you when it's available on demand and posted on laboratories.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.